Welcome to the June Radicle Community webinar. So my name is Timothy Byars. I'm the President and Chief Education Officer of Radicle Health, and joined today with Eloise Thiessen, our CEO. So these webinars are included in our Radicle Community membership package. They're free to all of our members. The Radicle Community is, it's comprised of advocates and educators and healthcare professionals, cannabis stakeholders, and really what we're looking to do is provide support, information, and resources to our members. So if you have if you want some information about the Radicle Community, you can go to our website, radiclehealthcare.com. Excuse me. Click on the community link, and you can find some more information about what else is included in addition to the webinars every month. I also want to remind everyone that we have an array of curriculum options at a range of price points for students and working professionals. So please reach out to us if you would like some more information about the courses that we offer, or you can just go to the website and click on the education link, and eventually you'll find yourself at our courseware website. So we have a bit of a shorter presentation today. It's only going to be about 30 minutes, but the content is really interesting, so I'm kind of excited about it. We're going to talk about cannabinoid emulsions, or cannabinoid nanoemulsions today. So everyone has likely heard the expression that you can't mix oil and water. And of course, this is an expression that is intended to convey an incompatibility between two things. So no matter how much you mix oil and water, they always eventually separate. But it's not really difficult to find examples of where oil and water do in fact mix and mix into a stable homogeneous substance. Milk, salad dressings, mayonnaise. These are all examples of mixtures that are called emulsions, and emulsions are simply mixtures of two or more liquids that are normally immiscible. So this means that the liquids typically can't form into a homogeneous mixture. So in an emulsion, there are very small droplets of one liquid, and they're dispersed into a second liquid. So if you've ever made a salad dressing using oil and vinegar, you've made an emulsion. And if you've only used those two ingredients, you likely witness that emulsion begins to separate very quickly. So the oil will form a layer on top of the vinegar. To create a stable emulsion in the kitchen, you need to use an emulsifier. Emulsifiers coat the small droplets in the emulsion, and it prevents the small droplets from coalescing, and it prevents the mixture from separating. So when you're making your salad dressing, you might have used, for example, an egg yolk to help stabilize the mixture and prevent it from separating. There are proteins in the egg yolk that include amino acids that both repel water as well as amino acids that attract water. So egg yolks are they're emulsifiers, and this is a basic emulsion. It's called a macroemulsion, and the droplet sizes are somewhere between like 100 and 300 micrometers. So a micrometer, 1,000 micrometers is equal to 1 millimeter, and you can see on the slide here, this is intended to represent a human hair. Micrometers are also called microns. So you can see up in the top left where the arrow is, that's 1 micron compared to the size of a human hair, and for some reason, dust mite feces too. So there are two other types of oil and water emulsions. You can also have water and oil emulsions, but we're going to talk about oil and water emulsions today. And those types are called microemulsions and nanoemulsions. And these types of emulsions are comprised of very small droplet sizes. They're much smaller than macroemulsions, and they are the reason why you can now drink your cannabis. So when formulating any cannabis medicine, there exists a lot of inherent challenges. So cannabinoids are highly lipophilic. They love fat. They degrade rapidly under oxidation. They have poor aqueous solubility, and they have poor bioavailability. So healthcare professionals and patients tend to like oral administration forms because healthcare professionals and drug formulators can facilitate the use of pharmaceutical grade ingredients and standard dosing protocols with oral formulations. And as patients, we're kind of used to taking medications orally. However, ingested cannabinoids, they degrade in the stomach, they undergo excessive first-pass metabolism in the liver, and of course they produce psychoactive metabolites. And often consumers fail to understand the delayed onset of ingested cannabinoids. And we all have heard the story. They consume more than they intended before the product takes effect. And then, of course, they have very unpleasant adverse effects. So the goal of cannabis drug formulation is to improve efficacy and stability and to reduce those variabilities. So, for example, the variability of onset during oral ingestion. And to address these challenges, there have been a lot of industries, the dairy industry, soft drink, juice industries, cosmetic industries, that have long used microemulsions and more recently nanoemulsions to improve things like clarity and weight and flavor of beverages and onset and bioavailability. The pharmaceutical industry has been using microemulsions to deliver oil-soluble active ingredients through the blood. Pesticides are often made using emulsions. They're commonly used in the cosmetics industry. Water can be emulsified into fuels to make them burn cleaner. And then sometimes oils are added into water-based beverages to provide some flavor. So what is a cannabinoid nanoemulsion? Broadly speaking, cannabinoid nanoemulsions are mixtures of cannabinoids, carrier oils, and other chemicals that make the mixture water soluble. And why do we need them? Remember that cannabinoids and terpenes are lipophilic, so they are fat-loving. They have low solubility in water. So in the human body, poor water solubility results in low bioavailability. So consequently, when you eat cannabis, your body only absorbs a fraction of the cannabinoids, and the rest are either metabolized into something else, or they're eliminated, or they're destroyed through some other process. If we can make cannabinoids more water soluble, then the body can absorb much more of the cannabinoid payload, and that increases bioavailability. Water solubility also increases time of onset, and we'll talk a little bit more about that shortly. So nanoemulsions promise to provide exceptionally high bioavailability. They can be taken orally, they can be applied to the skin, and again, they have rapid onset. Some manufacturers state that onset can occur in the mouth almost immediately upon oral administration. Don't know. For some consumers, this means higher potencies, faster onsets, lower doses, which means cost savings, and more product options because nanoemulsions are water compatible and they can easily be mixed into beverages at any concentration. So to summarize, cannabinoid nanoemulsions promise exceptionally high bioavailability, fast and complete absorption, water compatibility so they can be mixed into beverages, long-term stability. So there are some manufacturers who claim that their shelf life is as long, it can be counted in the years. And then you can have translucent and also transparent formulations. And these contribute to more appealing presentations, especially if you're mixing cannabinoids into water. So to discuss how nanoemulsions work, there are some terms that we need to define and understand, and those terms are bioavailability, surfactant, and micelles. So probably everyone on the call knows what bioavailability is, but we'll define it anyhow. It's the percentage of a dose of unchanged drug that reaches the bloodstream. So a drug that's administered intravenously has 100% bioavailability. When that drug is administered via other routes other than intravenous, its bioavailability decreases because there exists incomplete absorption, there is first-pass metabolism if you're ingesting it orally, and these are variables that are distinct in each patient. So all emulsifiers are types of surfactants, and the term surfactant is a portmanteau. It's a word it's a combination of three different words: surface, active, agent. And surfactants are compounds that reduce surface tension either between two liquids or between a liquid and a solid. So emulsifiers is a type of surfactant that reduces surface tension between two liquids. Surfactants have hydrophilic heads and hydrophobic tails. And when there are enough of these things enough of these surfactant molecules, they start to aggregate and they form structures called micelles. So you can see here in the slide, on the left-hand side, you have one surfactant molecule, and it's sort of sperm-like looking. It has a hydrophilic head, a water-loving head, and a hydrophobic tail, a water-hating tail. And so when there's a concentration of these in a mixture that contains oil and water, you can see on the right-hand side the way that they start to congregate. So all of the water-hating tails hide on the inside, or they're attracted to oil, and all of the water-loving heads form a structure on the outside. So a micelle is a spherical structure in which all of the hydrophobic tails of the molecules hide on the inside. They're shielded from water by the hydrophilic heads that line the outside. And here are just a couple of illustrative examples about what these things look like. We actually make micelles in our bodies. They're really critical for fat and vitamin absorption in the small intestine, and we're going to talk a little bit more about this later. So emulsions then, in theory, overcome the challenges of delivering water and soluble lipid compounds like cannabinoids. So we're not really going to talk anymore about macroemulsions. These, again, they're basic types of emulsions. They have very large droplet sizes. They're unstable. They don't improve bioavailability. They don't shorten time of onset. Microemulsions and nanoemulsions are sometimes used interchangeably, but there's actually a significant difference between them. And there's a reason why we're discussing cannabinoid nanoemulsions today and not cannabinoid microemulsions. So microemulsions can be transparent, they can be translucent. They're stable mixtures of oil, water, and surfactant. And they have droplet sizes they can be anywhere between 15 and 300 nanometers. So what's a nanometer? It's equal to 1/1000th of a meter, or 1/1,000,000th of a millimeter. I read somewhere that your hair grows at about 1 nanometer per second. So microemulsions and nanoemulsions, they're produced with somewhat similar processes, but microemulsions, it's the older technology, they require greater amounts of surfactant. So there's no specialized equipment that's needed when you're making a microemulsion. Chemists just simply overwhelm the water mixture with a lot of chemicals. So when these surfactants are put in with the oil and water phases, the micelles spontaneously form around, in our case, the cannabis oil and the carrier oil. Microemulsions do produce increased bioavailability. They do produce faster onset, but they require a lot of chemicals. And so this can lead to undesirable effects in the finished emulsion. So for example, if you were eating a microemulsion, it might taste like soap or detergent, or the levels might exceed maximum levels of human safety. So like microemulsions, nanoemulsions can be translucent or transparent. And again, they're stable mixtures of oil, water, and surfactant, and they have very small droplet sizes. So nanoemulsion-based formulations in which all of the droplets are smaller than 100 nanometers, these are translucent. And then if you can get the formulations between 15 and 50 nanometers, these are transparent. They have a high degree of clarity. Nanoemulsions can be made using far less surfactant than microemulsions because formulators use mechanical energy to break the droplets apart. Nanoemulsions are produced by utilizing ultrasonic cavitation-derived high shear forces. And this physically breaks the oil droplets into nanometer sizes. So what is ultrasonic cavitation-derived high shear force? I have no idea, and it's outside of the scope of this talk. But suffice it to say that the process uses high intensity and low frequency ultrasonic waves. So the takeaway is that it's mechanical. They're physically broken apart into smaller sizes. So when formulators are making nanoemulsions, they can use food-grade surfactants to make mixtures that are stable and, again, have long shelf lives. The ingredients that you would typically need in a nanoemulsion for a beverage, for example, would be obviously a cannabis extract, the carrier oil. Typically, this is a long-chain triglyceride, and we'll talk a little bit more about long-chain triglycerides in a minute. The surfactant. Usually they have some kind of preservative to prevent fungus or mold, and then water, and that's really it. So there's a company called Sono Mechanics, and they're a company that they develop and sell products to help facilitate formulation of nanoemulsions. But they have a website with a bunch of different information that's worth checking out. They suggest that a typical nanoemulsion concentrate might have the following. What you see here is the kind of the percentages of the ingredients. So you'd have about 5.5% of it would be the cannabis oil extract. A somewhat comparable percent of the mixture would be carrier oil, only 2% surfactant, and then 85% of the mixture is just distilled water. So if you were to make microemulsion, in comparison, a microemulsion would take about 50% of the mixture would be surfactant in a microemulsion. And the example here for nanoemulsions, it uses a level of surfactant that's well within regulatory compliance, and it doesn't compromise the beverage flavor. So maybe a couple of you are thinking, why do we need carrier oil? You know, why cannabinoids are lipids, right? They're fat soluble. Why don't the micelles just formulate right around the cannabinoid lipids? Why is the carrier oil necessary? It does two things. First, it helps stabilize the nanoemulsions. It protects them from oxidizing, especially if the formulators are using long-chain triglycerides. So when long-chain triglycerides are used in this manner, they're often referred to as ripening retarders. So they prevent oxidation and ripening. And then second, the carrier oil helps to maximize the absorption of the emulsion in the intestine. So for the highest bioavailability, we want the smallest droplet sizes. And emulsions can achieve the smallest droplet size when the dispersed phase, which is in this case the oil, and the continuous phase, the water, have similar viscosities. So we all know what cannabinoid extracts look like. They're really viscous and sticky, much more so than water. So the carrier oil helps to reduce the cannabinoid extract viscosity, and it brings it closer to that of water. And so it helps to produce a mixture with smaller droplet sizes. So for oral ingestion, if you're going to be eating long-chain triglycerides, it's got to be food grade. It can't compromise the appearance or taste or texture or stability of the end product. And that's often why formulators are using long-chain triglycerides, because they can form a hydrophobic core that's large enough to accommodate the cannabinoid payload. So remember, cannabinoids are rather long molecules. Medium-chain triglycerides are actually more water-soluble, in some studies up to 100 times more water-soluble than long chains, but they have pretty small cores. And even though they can hold cannabinoid payloads, they don't do it as effectively as long-chain triglycerides. So why do these droplet sizes produce higher bioavailability and faster onset? When you consume any oil or fatty substance, it arrives in your small intestine as large drops. And your small intestine is covered by epithelial cells. Remember, we talked about this during our COVID talk. You know, epithelial cells line our entire mucosa from front to back, and they're intended to keep the inside in and the outside out. So when you eat fats, to get absorbed into your intestine, they need to cross the aqueous layer of your epithelial cells. So what happens when you eat a fat is that your gallbladder starts to reduce a bile, and that bile contains phospholipids, and it creates an emulsion. Remember that phospholipids, they look like our surfactants. They have that, that hydrophilic head, hydrophobic tail. So once these phospholipids are produced out of this bile from the gallbladder, micelles are formed. There's a concentration of them. They start to engulf the fats or lipids. And these micelles that are formed are between 10 and 20 nanometers in diameter, and they're able to carry the fatty compounds through the epithelial cells because they're small, they're physically small enough. So these fats that we eat, they are able to absorb into the small intestine, but it takes time. And the process also competes with metabolism, and it also competes with elimination. It also competes with other destructive processes. In fact, this is the reason why when you eat an edible, it takes about an hour for it to take effect, because that's the time it takes for these micelles to form, for these cannabinoids to get absorbed into your small intestine, and then also for your liver to metabolize whatever's left over. If you just consume a nanoemulsion with the micelles that are preformed, the payload is pre-digested. So as soon as it arrives in the small intestine, it can get absorbed. The size of the droplet and the carrier oil both play an important role here. So long-chain triglycerides demonstrate highest bioavailability after oral administration. So examples of long chains include things like olive oil and corn oil, fish oil, and then that's followed by medium chain triglycerides, MCT. This is usually fractionated coconut oil, and then non-triglyceride oils, things like mineral oil. Again, long chains result in better intestinal transport because they form micelles that are slightly larger than those that are formed by MCT, still small enough to get absorbed, but larger enough to hold those larger payloads of cannabinoids, and so you get better bioavailability. So one thing we like to ask at Radicle Health is there any evidence of that? So there's certainly evidence that nanoemulsion technology does improve bioavailability of a drug. It can be non-toxic, it can be non-irritant in nature, it can improve the stability of drugs, and it can improve the absorption of drugs. So we've seen a lot of evidence in the pharmaceutical industry alone using microemulsions and nanoemulsions to help drugs with the onset and to help with the bioavailability. There's not, however, a lot of evidence demonstrating that there's safety and efficacy specifically with cannabinoids. In fact, I could find only 2 studies. So if anyone else has any studies that they know about after this presentation that I'm not talking about, please send me a link. I'd love to look into it. But these are the 2 that I could find. So the first one was published in 2019. It looks specifically at the effects of a CBD nanoemulsion in a rat model. So these researchers created a nanoemulsion. It was 99% pure CBD powder. Then the oil phase was vitamin E, ethanol, and tween 20. So tween 20 is polysorbate 20. It's a common surfactant that's used. And then distilled water. The researchers separated the rats into 2 groups. They orally administered a CBD oil, just a regular CBD oil, to one group, and then they gave the nanoemulsion to the other. And then they collected blood samples over a 24-hour period to examine the absorption. So for time of onset, it's difficult to ask a rat how it's feeling. So they measure this by testing Cmax, or this is the maximum serum concentration in the plasma. So the rats absorbed the nanoemulsion much faster than the CBD oil, and the peak effects were measured at 2.5 hours for the nanoemulsion versus 8 hours for the CBD oil. That's an improvement of a factor of 3. That's really significant. What about bioavailability? So we should note first that absolute bioavailability of CBD in rats is way higher than it is in humans. So it's probably due to lower first-pass metabolism in rats. But there have been other studies, previous studies, that have measured absolute bioavailability of CBD. It's established at about 70%. In humans, it's roughly about 6%. But in our study here, the bioavailability of CBD oil was measured at 73%, so right in that target. The bioavailability of the nanoemulsion was nearly 94%. So again, another significant improvement. The other thing that the researchers determined in the study is that the CBD oil relied on bile-mediated micelle formation. So remember, when you eat fats, your gallbladder produces a bile. That bile has phospholipids, and those phospholipids create an emulsion, and eventually the concentration creates micelles, and those micelles carry those fats through the epithelial cells, and that takes a lot of time. If the CBD oil relies on bile-mediated micelle formation, that accounts for time of onset, right? That's the 8 hours for the Cmax. They determined that the nanoemulsion did not rely on bile-mediated micelle formation. So the nanoemulsion was likely absorbed directly into the small intestine. And again, that's why we see an improvement of Cmax by a factor of 3 with the nanoemulsion. And then finally, researchers stated that the appearance and particle sizes of the nanoemulsion remained stable after 6 months of storage. They only kept it for 6 months, but at the 6-month period, it was completely stable. So there is a second study, which was actually done in humans. Researchers compared the bioavailability of a nanoemulsion with that of Sativex in 14 healthy volunteers. So this was done in 2018. These researchers created a novel oral THC and CBD nanoemulsion. They called it PTL 401. And then the volunteers received either a single dose of PTL 401 or an equivalent dose of Sativex. Now remember, Sativex is formulated as a transmucosal drug. It's intended to have a faster onset and higher bioavailability than other oral delivery formulations. So the Cmax for the nanoemulsion in this study was 1.6 times higher than that for the equivalent dose of Sativex, and was for both THC and CBD. The nanoemulsion time to maximum concentration was significantly shorter for both CBD and THC, so 1.3 hours for the nanoemulsion versus 3.5 hours for Sativex. So this was another study where the researchers noted that the nanoemulsion demonstrated an increase in bioavailability for both CBD and THC, and they concluded that this relatively faster absorption and improved bioavailability compared to Sativex justifies further larger scale clinical trials with this formulation. And for the research, that's really about it. So, you know, I'm always curious to hear from you guys if you're using or recommending these products to your patients, or if you've ever tried them, what your experience has been. Before we end, I just want to go over briefly how a cannabis beverage is made, because it's kind of interesting. And you can see there's a workflow diagram here. This is also from the Sonomechanics website. You can see that there are two distinct workflows, one for each of the phases. The one workflow is for the dispersed phase or the oil, and then the second one is for the continuous phase or the water. So in the oil phase, you see that there's cannabis. It's extracted using supercritical CO2. It's winterized and decarbed. Then they add the carrier oil. In this case, they're listing both long chain and medium chain. There's a little bit of surfactant added to this phase, and then it gets premixed and it sits. And then concurrently, there's the water phase, which is really just water and surfactant, and that mixes for a while. And all of this stuff then goes into a main tank. It goes through its ultrasonic cavitation, high density, break stuff apart thing, and then they filter it, and then it goes into bottling. And the bottling is kind of interesting, because the final step of formulation is they do a sterile filter, and then they transfer it into a light protected container. But the formulators use a hydrophilic filter membrane that has a 220 nanometer pores. And that removes all the contaminants. So the microorganisms and dust and plant matter and other contaminants formed during the homogenation process are physically filtered out because of the size of the filter. That's generally how filtering is done. But the nanoemulsions, the emulsions are comprised of droplet sizes that are smaller than 100 nanometers. So these droplets can pass directly through the filter membrane. And they also use a hydrophilic membrane. So that ensures that the droplets don't stick to anything. And then prior to capping, you can see this box that says LN2, that stands for liquid nitrogen. They might add a little bit of liquid nitrogen to dispel any oxygen and to pressurize the content in the bottle. Now, expelling atmospheric gases prior to bottling, using things like heavy inert gases is a practice that's used in other industries also. For example, it's used here in California quite a bit in the wine industry. We used to use argon, for example, to dispel any oxygen. It's got to be heavier than oxygen. But when these types of things are added to the container, the liquid nitrogen, for example, will rapidly expand as it changes from a liquid to a gas, and then it pushes the oxygen outside of the container. And then they package it up and ship them, and then they're ready for us to consume. So just in closing, I think that there are a couple of really interesting things about nanoemulsions. And I'll preface this by saying we still have a lot to learn about these things. We still have a lot of data that we need to derive from these products. It's, you know, like everything in the cannabis industry, it's a little like putting the cart before the horse. You know, we can predict that these things are safe based on their uses in other industries, but we really need more data here in the cannabinoid industry to make sure that these are safe. But there are a couple of interesting things about these products. First, they've created a new kind of a new route of administration, or rather they're combining different facets of two different routes of administration. So healthcare professionals can now use oral administration, but have a time of onset and a duration that's similar to that of inhalation. And, you know, it has incredibly higher bioavailability. So you take these, you know, the sort of the best attributes of oral administration and inhalation, and you combine them into this new route of administration. And this provides a novel approach for clinicians and patients to use and to treat disease. And again, you know, as a patient, we're kind of used to consuming things orally. So it's a really interesting prospect. And then secondly, these products, I think for the first time, create sessionability with cannabis products. So for people who kind of want to hang out with family and friends for a couple of hours, this experience is very much like sharing a bottle of wine or a couple of beers. If you select low dose products, they're going to take effect, presumably, within a few minutes, maybe 15 minutes. They're only going to last a couple of hours, and you can moderate those effects by moderating your consumption in the same way that you do with alcohol. Now, I personally haven't used any of these products yet, so I can't speak from personal experience. But you know, this is what the data says. So I'm excited to try these products. I hope that they do have these kinds of effects, where there's fast time of onset, and the duration is much less, because you're not getting the metabolism into 11-hydroxy, which can last 8 hours. So I want to thank you guys for attending today and for indulging me, and I'm hoping that we have some questions or comments. So go ahead and unmute yourself, and let's ask some questions or have a discussion.
That was great, Tim. I know that personally what I've experienced with myself and with some patients, now I don't use nanoemulsion or nano products very often, because I'm not, well, up until this point, I wasn't really sure how they were processed and whether or not they were considered safe. So I have noticed a higher bioavailability, so people can take smaller dosages, but the onset's still incredibly variable. And I've tried one drink that I did notice worked a little bit quicker, and there's definitely more reports of, you know, cannabis drinks having quicker onset. So I'm curious to try them as well in terms of seeing if we get better results there, but they're incredibly expensive. I mean, I just saw an advertisement the other day for like a 6-pack of these cannabis fizz drinks. It was $27 before taxes. So you can assume after taxes, you're going to pay about $40 for a 6-pack, which is about $7 a can, which I guess could be like having a cocktail if you're out and about, right? It's like about the price of a beer.
Yeah, this bottle of Artet, which is a cannabis aperitif, so it's intended to be just maybe drank over ice, for example, and you can control the dose just by the volume that you pour. It's $56 before tax. So you're gonna get this. It's basically like getting a bottle of wine. It's 750 mils for just south of, you know, probably $90, maybe closer to $100 by the time you get out the door.